The other type of endoplasmic reticulum is smooth endoplasmic reticulum. SER cisterna more is tubular, not as a sac, and these channels interconnected together in various shapes and various size. SER involved in the phospholipid and steroid synthesis enzyme, such as in the cells in the adrenal cortex. In some cells, such as the liver, they are involved in the detoxification, that the liver cells include the one type of protein that is a P4050, 450, that the liver cells include some enzymes that are involved in the oxidation, conjugation, and methylation of some drugs and others. And in some cells, such as striated muscle cells, SER store and release calcium. But the main function of them is the production of the lipid and phospholipid of the cell membrane. These phospholipids that synthesis in the SER transfer to other membranous organelles, such as cell membrane, RER, stem cells, mitochondria, Golgi, that all of them are membranous organelles. They transfer by two, one of them by the formation of the vesicles, that the vesicle detach, move, and again fuse to another membranous organelle. And in some times, maybe it's carried by some phospholipid transporting protein charge, that they act as a carrier and transfer the phospholipid from the SER to cell membrane or other membranous organelles. For example, here the membrane of SER is rich in the phospholipid, that this phospholipid transferred by these carrier to other membranous organelles that are poor in the phospholipid membrane, and then again it come back and start with carrying the phospholipid. Golgi apparatus is the other organelle, other membranous organelle in the cell, that its name as also as shipper. The main function of the Golgi apparatus is some post-translational modification, such as glycosylation, sulfitation, or others occurred in the Golgi apparatus, and also package and addressing proteins that synthesis in the RER. The Golgi apparatus transport material to different locations in the inside or outside of the cell, and its appearance is as a stack of the pancake. It includes some cisterna that one of them is toward to the RER or SER, and other one is toward to the cytosol or cell membrane, and between of them there are other cisterna. The cisterna that is toward to the RER or SER named cis phase or reception phase. That this is this part of Golgi can some vesicles bubbling from the RER or maybe from the Golgi apparatus for retrograde transportation. These transport vesicles that are newly synthesized proteins in the RER transfer to the Golgi apparatus from cis phase for other process, such as post-translational modification, or maybe for packaging and transfer to other part of the cell. The last one, cisterna, named transphase or transfer phase. That in this phase, mostly include the large circles that name condensing vacuole, that the materials accumulate in them and form the secretory vesicle, that these vesicles separate from the transphase and transfer to other part of the cytosol or other part of the cells. And the cisterna that located between trans and cis phase are the intermediate or medial cisterna that mostly involved in the post-translational modification. For the transfer of the vesicles between the Golgi and ER, or also cell membrane or the other part of the cell, involve the two types of proteins that name coated protein type 1 and coated protein type 2. As you hear schematically, COP1 involved in the retrograde transportation from Golgi apparatus to ER, and COP2 involved in the transformation of the vesicle from ER to Golgi apparatus. And finally, the vesicles that matured and should be transferred from the Golgi apparatus to other part of the cell, such as lysosomes, mitochondria, or the cell membrane, separate from the Golgi and transfer to other parts. The other organelles are vesicles. That vesicles are membranous organelles that may be formed by the endocytosis from cell membrane, or maybe exocytosis from the protein synthesis that we produce in the ER, or maybe Golgi apparatus. That there are different types of vesicles in the cytoplasm that are restricted by a cell membrane. One thing that I should explain for you and notice you to it, that the structure of the membranous organelles, the cell membrane of these organelles structurally are similar to cell membrane. All of them have two layers of phospholipid and also integral and peripheral proteins that are general in all of the membranous organelles. Other organelle is a mitochondria. The other name of it is a powerhouse that involved in the energy formation, breakdown food to ATP, and ATP is a major fuel for all cell activity that require to energy. Mitochondria is an organelle that have double membrane. That these two membranes separate from together by intermembranous space. This is a outer membrane, inner membrane that separate by intermembranous space, and in the internal part of the inner membrane is a intermembrane space. Sorry, the innermost matrix, and here is a narrow intermembrane space. The outer membrane of the mitochondria is smooth. It includes some transmembrane protein that named porine. Porine is a type of transmembrane protein that are in the outer surface of the mitochondria that transfer small molecules to intermembranous space, and also involved in the releasing of some materials from intermembranous space to outer part. For example, cytochrome C is one type of protein that are in the intermembranous space, that involve in the apoptosis. When apoptosis activate by intrinsic pathway, the cytochrome C released by this foreign transmembrane protein. This membrane is smooth against of it. The inner membrane of the mitochondria have some enfolding to matrix that named crystal. Mitochondria have unusual phospholipids that highly impermeable to ions. And also, in the surface of the crystal include the multimeric protein complex that resemble to globular unit on a short stalk, that these globular particles involved in the ATP synthesis in most of the cells. The crystal increase the surface of the inner membrane, and the size and the length and the amount of them can change when the cell is active or in the passive condition. And also mitochondria now divided and produce more mitochondria, that when the cell need more mitochondria, they can do by itself. In the matrix of the, in the inner membrane, additional to the particles that synthesize the ATP, there is one type of transmembrane protein that's named thermogenin, that this thermogenin involved in the heat production. It means that the mitochondria have two functions: ATP synthesis and also control of the body temperature by this protein that produce the heat by influx of the proton to inside of the mitochondria. Mitochondrial matrix that's located in the inside part of the inner membrane is a gel that containing numerous of enzymes and also DNA, ribosomes, and messenger RNA and transfer RNA, that a little amount of the protein synthesis occurs in the small population of the mitochondria. But most of the DNA, RNA, and protein of the mitochondria synthesis in the cytoplasm, and after that transfer to mitochondria. In this part, you know about the cytoplasm. That cytoplasm is a material that within the cell enclosed by cell membrane, except the cell nucleus. The main component of the cytoplasm is cytosol, that is a jelly-like substance that includes the organelles and inclusion. That in this part, you know about some organelles, and in the next part, I explain other organelles that are in the